audible? Thank you. So, just to begin with the presentation, I would like to thank Advent team, Surajit and entire team for giving me the opportunity to present our work here. Nonetheless, Molsoft team, I think we are the first one in India who actually got Molsoft way back in 2010.
Yes.
And the entire team of Molsoft, Andrew, Eugene, Aurigene, and then Max, entire team of people who supported us over the years. And we identified that point of time that this is the kind of software which actually is suitable for our kind of environment, which is light enough, which is modifiable, which can be developed in multiple areas. And that is where we actually switched. My colleague Suvendu will agree with me. When we were together, the same team working together at Aurigene. Let me briefly give you an understanding about Aurigene Pharmaceutical Services and Aurigene Oncology. We are two different companies. Oncology is the innovative company who is taking their compounds to clinic. We are as a service provider who are working with several other partners purely in service mode. Okay, so many of the work which we did together with Suvendu and our team at Aurigene, we cannot present it. And something which we do it later on, most of them are belonging to clients. As you know that when you work in service industry, you cannot present many things. You experiment so many things, but we cannot present those. So I thought, why don't we put together a small case study in-house to show our capability in terms of the design, synthesis and testing with quick turnaround time. So that's why we choose one of the target that is called RIPK-2 kinase as a target for degradation, and objective was to showcase how effectively we can use all the available techniques which is available in Molsoft, in-house technology, as well as the in-house lab synthesis tool as well as testing tool to rapidly address some of the problem which is faced in this degrader field. So with that, actually I'll switch to something which I want to highlight. This is something different, non-science. So before I start this presentation, I want to bring your attention towards this one. How many of you can recognize what is this? Anybody knows this bird's name? Exactly.
Uttarakhand state bird.
Exactly. Yes. So this is the state bird of Uttarakhand, national bird of Nepal, called Monal, which I took this photograph at 13,000 feet height. Whenever you go to Himalaya, please do visit those places. India is beautiful. So this photograph was taken at 13,000 feet height and one of the beautiful birds. And let me tell you, science is nothing different from this beauty. The more you do it, you enjoy it. And that is where the beauty lies. So this is the passion which is driving us towards something innovative, doing something innovative in science as well. So that's why we challenge ourselves. Now, this is one of the beautiful birds I have ever seen and took these photographs at that height. Anyway, so this present work, which is a disclaimer, it is the work done at APSL, that's called Aurigene Pharmaceutical Services Ltd. And some of the materials are distributable, some things are not, but majority is disclosed to educate people that how can we integrate many things to address some of the challenges what we face in day-to-day life in molecular modeling. So first, let me acknowledge my team who are working in this project, our senior management team, medicinal chemistry team, my team, entire team who are there here, present members as well as past team members. Siva, Suresh, Madhuri, they are not there at this present in my company, but they were working on this particular projects long time. Manvi right here, Sijan, Resmi, they are all here. They are also working this project. They are the one who championed this project. Our other team, in vitro biology, medicinal chemistry, as well as IPM. So it's an integrated project altogether. It's not only one. Thank you, team. So my job became easier after Suvendu and Dr. Ramesh presented all the plethora of work towards the PROTAC development. I don't have to conceptually make you understand what PROTAC is all about. But let me tell you something which has come up recently. Now, the first compound to be approved soon is phase 3 development at the stage of phase 3 now. So the field has evolved from 2000 to 2023. With that, actually there are different approaches, PROTACs and glues. Our efforts at Aurigene is both. So we work on PROTAC, we work on glues also. We have developed methods to address PROTAC challenges. We have developed methods from the ground up approach from the scratch to address challenges towards glue. So these are our typical problem for the experiment as well. And the way PROTAC is developed for anything, similarly glue is equally challenging. So I'll give a glimpse of the approach what we have done for PROTAC, but glues we have expertise on it and we have delivered some of the compounds designing novel glues also. So with monovalent and bivalent there are typical differences. Most challenges goes with the design as well as not only designs, its developabilities towards later stage. So now monovalent means the glues, bivalent is the PROTAC. They deviate certain rules from the small molecule and also they the PROTAC, they deviate certain rules from the small molecule. There's pretty good recent paper. This was 2024. Last week, this paper was published by Crews, the same group who are pioneer in PROTAC, and excellent work they have just done over the decades. I'll not go to different type of PROTACs, how LITACs, all these things, but a summary of various approach, covalent, homoprotacs and all, these are all there in the public domains. You can go through those reference literatures wherein you can think about some of the target degradations towards the different targets towards intracellular proteins, as well as some effects, some efforts towards the extracellular target proteins also. So there are a lot of efforts going on. This is an evolving field from the experiment perspective as well as the computational perspective. Now, the challenges what I want to focus here is mainly the properties. Like we are thinking about a compound for treating some disease, not designing some molecule in the lab. So essentially the challenge with PROTAC generally is the PK, PD, then specific to the degradation activity, their warhead design, complex formations, and so many other things. For example, hook effects, and ultimately the oral bioavailabilities. So these are the practical challenge which is faced for any PROTAC design and development. Now, with that background, I'll just switch over to my case studies where I'm presenting about RIPK-2. This is one of the kinase target again, very friendly for modeling and as well as some of the, but the biology is completely different. So every kinase, although research has been done since long, I've been working for kinase targets since 2000. So same target which I used to work there, now also it's clinically relevant. So a target is never absolute. Always it is there in the field to something to be done in those targets. So this is one of the target where we have worked on called RIPK-2, which is pretty much involved in some of the pathways related to the inflammation as well as oncology related pathway. So our objective was very simple. First, how can you speed up the process of design? Challenge to address low permeability, metabolic stability, and if you can do that one in a sophisticated methods or automatize many of the things in such a way that we can efficiently deliver or design to the lab in a quick time so that they can synthesize it and test it. So this is some of the challenge every company face. As a service provider, we should be first to provide this kind of solutions in a quick fraction of time. So final results what we could do is that within a short period of time we could identify potent degrader with suitable ADME and PK properties. However, the PK and ADMEs are not that optimum to go for the oral administrations, but we could demonstrate that, okay, this is working, so that it can be applied in projects. So, as usual, same methods and protocol. So, we have a set of partial PROTAC library in-house. Medicinal chemistry, we maintain all the partial PROTAC library for several E3 ligase along with the linkers. Predict the ternary complex, improve the potency in terms of the sampling as well as the linker length as well as POI designs. So here we do extensive PUs of MD simulations as well as complex formations. So we use a range of methods, not only sticking to ICM but range of methods to apply this. We use the DNN models for predictions of degradations, followed by generations of PROTAC using generative methods. We design compounds, novel protein, the ligand for protein of interest using our AI platform, wherein we have a large amount of data to generate novel compound for any specific targets. Then iteratively we use those data to design. Then our synthetic team, they synthesize the compound. Then we do kind of testing and also incorporate the data back to our, again, design cycle. So this entire cycle of designs, synthesis and testing for this project we finished within 2 months timeline. So whole gamut of PROTACs library what we have in-house, which we actually quickly take as a concept basis first. So basically when we start any projects, whether any concept is working or not, through your modeling as well as your lab work should be tested first lot. So quickly we generate all this library with so many designs iteratively to mature compounds, mature PROTAC, and then test it with a ternary complex, then follow the cycle and prioritize further. So we did the same thing for this particular project within short period of time. Then we model the ternary complex using the ICM tool. We use DNN model to predict the DC50 and Dmax for all the design compounds. Give that ideas that these are the priorities compounds. We have to, priority PROTAC compounds, we have to prioritize and faster the synthesis. And here is the cartoon diagram wherein you see RIPK-2 and CRBN complex, ternary complex, along with the hidden structures. Some structures I cannot show that. Some structures which was pretty much giving that kind of potency. So these are all predicted value first and shortlisted all this library. Once we got those data, this is the set of around 8 or 9 compounds what we did the first lot. Within the first lot itself we could see RIPK-2 potency was retained. Then with respect to the binding energies and all related to Molsoft, that is calculated. But this is the experimental data which was obtained after we designed the compound synthesizing and testing. So you can see that a lot of 500 or so library of designs. From that we could shortlist around 10 or 15 compound library and prioritize those one for synthesis. So that is a compound called 33P, which I will be discussing in the next slide. So typically we compare the small molecule versus the one of the degrader to understand whether there is a degradation effect. So here actually we could able to see that the degrader has very good potency equally maintained with respect to the inhibitor, which is around 3 nanomolar compared to 9 nanomolar, and their degradation values are also shown here, and it was pretty well established. With respect to the permeability and metabolic stability, this is the first challenge what we saw in the project, wherein although we attend all the degradations as well as the potency, wherein we saw these compounds typically case of PROTAC, low permeability, there would be metabolic stability issues. So here in first compounds wherein we saw that low metabolic stability and permeabilities, in fact they are clean in SIP and other properties, but you can clearly see that stability was an issue. Now same thing we can, after 1 or 2 modifications, we obtained the compound 33, which was equally degrading, but it was not sufficient. So possible reasons of hook effect probably. So with that actually we could attend this compound with a decent IVPK as well as CPK, which was something which we could have able to optimize from that. And herein I can add one point wherein we have established the assays, for example EPSA, which is experimental polar surface area calculations, which is experimentally determined. So our assay is pretty robust, which can calculate the polar surface area which is related to the permeability of beyond rule of 5 compounds. So that information is already there with compounds when we synthesize. So we tend to incorporate those data to these design parts wherein we can address the permeability and other factors. Now this is the IB and SCPK, poor. I can agree it's poor data. But within the showcase that within the short period of time we could able to establish some of these methods to prioritize compound and reach to this stage within the short period of time. That actually resulted in the some compound which is little bit having exposure towards administrations in IV route or ISC route. So with that actually I'll just finish my presentations with entire gamut of technologies and platforms what is available at Aurigene Pharmaceutical Services to efficiently support the degradations projects. So our biology understanding starting from CAD-based design, then synthesis, testing, entire gamut of assay, everything is available. So this gives a kind of close, a kind of speed up in terms of design and iterations towards the synthesis as well as testing cycle. With that, I conclude. Thank you for your patience.
Thank you, Dr. Sunil, for such a engaging and impactful session.